Welcome to this presentation on the development and growth of limbs. Limb development is a cornerstone of developmental biology, offering profound insights into pattern formation, morphogenesis, and cellular differentiation. In humans, this remarkable process begins in the fourth week of gestation and continues through the eighth week, laying the foundation for the appendicular skeleton and its associated tissues. It involves intricate coordination of signaling pathways, transcription factors, and cellular behaviors. Simple mesenchymal cell populations transform into complex functional limbs through precise spatio-temporal mechanisms. This video will explore the key stages of limb development from initial specification to the formation of bones, muscles, blood vessels, and nerves. It will also examine developmental abnormalities and their clinical significance. Join me as I unravel the fascinating journey of limb formation and its implications for science and medicine. Limb development starts with the precise specification of limb fields along the embryo's anterior-posterior axis. The upper limb field forms at the level of HOC6 expression, aligning with the first thoracic vertebra, while the lower limb field emerges more posteriorly. Retinoic acid signaling from Hensen's node is pivotal, activating specific HOX genes to establish these positional identities. The T-box transcription factors TBX5 and TBX4 are critical for this process, driving forelimb and hindlimb specification respectively. These factors determine limb type identity, guiding the development of distinct structures such as wings versus legs in model organisms. This early specification sets the stage for subsequent limb bud formation and patterning, orchestrating the complexity of molecular signals that shape functional limbs. Join me as we explore how these initial steps lay the foundation for the remarkable process of limb development. After specifying limb fields along the embryo's anterior-posterior axis, limb bud formation begins the next essential stage in human development. This process starts around day 26 for upper limbs and day 28 for lower limbs. Mesenchymal cells from the lateral plate mesoderm proliferate rapidly, forming a distinct bulge under the overlying ectoderm. FGF10, secreted by the lateral plate mesoderm, serves as the primary signal for limb bud initiation, establishing a critical feedback loop with FGF8 expressed in the induced ectoderm. This FGF10, FGF8 signaling cascade sustains limb bud outgrowth and ensures its structural stability. The early limb bud comprises a mesenchymal core enveloped by ectoderm, with the progress zone housing rapidly proliferating undifferentiated cells. These cells lay the foundation for subsequent patterning, enabling the development of bones, muscles, and nerves. Let's explore how these buds evolve into the intricate structures of functional limbs. Following limb bud formation, limb development organizes along three principal axes: proximodistal, antero-posterior, and dorsal-ventral. The proximodistal axis, extending from shoulder to fingers, is regulated by the apical ectodermal ridge through FGF signaling, guiding outgrowth from proximal to distal structures. The antero-posterior axis, running from the thumb to the little finger, relies on sonic hedgehog signaling from the zone of polarizing activity to establish digit order. The dorsal-ventral axis, distinguishing the back from the palm of the hand, is controlled by Wnt7a in the dorsal ectoderm and engrailed 1 in the ventral ectoderm, defining the hand's orientation. These axes operate in a coordinated manner with extensive crosstalk among their signaling pathways to ensure precise limb patterning. This system transforms the early limb bud into a structured appendage, setting the stage for tissue differentiation. Let's examine how these axes shape the complex architecture of functional limbs. Building on the three-axis patterning system, limb development relies on three signaling centers: the apical ectodermal ridge, the zone of polarizing activity, and the dorsal ectoderm. The apical ectoderm ridge secretes FGF4, FGF8, and FGF9, driving mesenchymal cell proliferation and delaying differentiation to ensure proper growth. The zone of polarizing activity produces sonic hedgehog, which defines digit identity and number while sustaining ridge function through a feedback loop with gremlin and BMP signaling. The dorsal ectoderm contributes to patterning via Wnt signaling, shaping limb orientation. Recent studies highlight additional mechanisms such as membrane depolarization and calcium signaling, which triggers chondrogenesis. These centers coordinate through complex networks involving BMP Wnt and Hox gene interactions, guiding the limb bud toward its final form. Let's explore how these molecular pathways work together to shape the development of functional limbs. Following the coordination of signaling centers, chondrogenesis marks the beginning of skeletal element formation and limb development. Around day 33 in humans, mesenchymal cells within the limb bud aggregate into precartilage condensations. These condensations, shaped and sized by SHH and FGF gradients, form the blueprint for future skeletal structures. The transcription factor SOX9 is central to this process, sustained by BMP signaling and directly controlling cartilage-specific genes like type 2 collagen and aggrecan. These genes drive the production of cartilage matrix, forming the foundation of the skeletal framework. Recent research tells us about the role of membrane depolarization and L-type voltage-gated calcium channels, particularly CAV 1.2, in regulating SOX9 expression to ensure proper chondrogenesis. This process transforms mesenchymal cells into cartilage, paving the way for bone development. Let's explore how these mechanisms guide the formation of the limb skeletal structure. After cartilage forms the skeletal framework, muscle development begins in the limb bud. Around day 36 in human development, myogenic precursor cells migrate from the somites into the limb bud. Recent studies reveal two distinct phases of myogenesis, embryonic and fetal. The embryonic phase involves PAX3 positive progenitors differentiating into primary muscle fibers, laying the initial muscle structure. The fetal phase follows, with PAX7 positive progenitors forming secondary muscle fibers for further growth. Wnt signaling plays a key role, inducing PAX3 to specify myogenic precursors and regulating myogenic factors, MyoD and MyF5, to drive muscle differentiation. The transcription factor mucilin, also known as MSC, is critical for maintaining muscle stem cell identity. These processes ensure the formation of functional limb muscles. Let's explore how these mechanisms shape the muscles essential for movement and strength. Following muscle development, vascular and neural systems shape the limb's functionality. Around the 4th week of human development, the limb bud relies on capillary network for blood supply. This network consolidates into a main stem artery, draining into a marginal vein to support growing tissues. Concurrently, peripheral nerves originate from the neural crest and migrate into the limb bud. These nerves extend axons guided by trophic cues like efferents from muscles and connective tissues. Chemoattraction and repulsion mechanisms establish precise nerve-muscle connections critical for limb function. Timing is vital, as early limb movements starting around day 42 drive proper bone and neuromuscular development. These coordinated vascular and neural processes ensure the limb becomes a functional unit with robust circulation and innervation. Let's explore how these systems work together to enable movement and sustain the developing limb's complex needs. After vascular and neural systems develop, congenital limb defects present significant challenges. These abnormalities affect roughly 1 in 1900 to 2100 newborns. They fall into several categories: longitudinal deficiencies impacting specific bones, transverse deficiencies resembling amputations, failure of separation causing syndactyly, duplication leading to polydactyly, and growth abnormalities altering limb size. Recent studies pinpoint genetic causes such as mutations in Hox genes, which are critical for limb patterning. These mutations can result in digit malformations or limb truncations. Environmental factors also play a role. Thalidomide exposure, amniotic band syndrome, and maternal diabetes disrupt normal signaling pathways, leading to defects. Understanding these causes enhances our ability to address these conditions. Let's explore how genetic and environmental factors converge to influence limb development and contribute to these complex abnormalities. Following our exploration of limb defects, understanding limb development holds immense clinical value. It informs treatments for congenital anomalies, advances regenerative therapies, and enhances prosthetic technologies. Recent discoveries have identified molecular signatures that convert non-limb forming cells into limb progenitors, paving the way for regenerative medicine. Single-cell and spatial transcriptomics have revealed new cell populations and regulatory networks, deepening our knowledge of human limb development. Ongoing research emphasizes stem cell therapies, tissue engineering, and bioengineered limbs to address limb disorders. By integrating developmental biology with clinical practice, we improve prevention, diagnosis, and treatment, enhancing outcomes for patients with limb deficiencies. This journey into limb development tells us about its transformative potential for medicine. Thank you for watching. I hope you enjoyed learning about limb development.